next section in biotech is biotech in human health. And this is looking at the treatment, cure, and prevention of different diseases. We're going to take a look at each of these. So we're going to take a look at how we produce medications to treat diseases. This is where we have the most kind of traction in biotechnology as far as human health. We're going to look at curing diseases. That's where we're working on it, but we don't have very much as far as, cures right now for different genetic diseases, and we'll talk about why that is. And then we'll talk about preventing the diseases from occurring in the first place. And we have some different ways that we can do that at this point. So we're going to take a look first at the treatment of diseases. So medicines, how do we produce different medicines using biotechnology? And the first one is looking at recombinant DNA technology. In fact, that's how we're producing medications of different types using biotechnology. So why do some bacteria produce human insulin? And my first question is, why would we need to produce human insulin? So who needs insulin as far as medication goes? People who have diabetes, right? So people who need to monitor their blood sugar and then adjust their blood sugar using insulin are going to need insulin. Specifically, human insulin is better if we can use it. We also have insulin from things like cattle, beef, and other sources, but they don't work as well for a lot of people. And until recently, they were fairly expensive. I know medications are expensive in general, but the production of human insulin made a huge difference in the market using this recombinant DNA technology. The idea is that we take bacteria and we take our human pancreas cell. We can take any cell, but we focus on the pancreas cell because that's where the gene is active. So we go in and we find the insulin producing gene in those cells that's turned on. And we take a plasmid, which is one of those sections of DNA, combine it together with that human insulin producing gene, then introduce that whole section of that plasmid, that completed plasmid with the insulin producing gene and the extra section from the original bacteria into a new one. Then we make these bacteria very happy. And we do this a bunch of times. We don't just do it with one, we'll do it with a bunch of them, but then we make them incredibly happy in a fermentation tank. So we're going to make them warm, we're going to give them food, and they're going to multiply. So they're going to make copies. Every time they multiply, they make copies of this plasmid, which has this gene that they're going to read. So they will read that gene and make that product. And so you can see here, they've zoomed in on one. Those bacteria are going to make human insulin as they're sitting there in that fermentation tank. So we eventually can extract and purify that human insulin and use that for market. So this is one of the first ways that we use this recombinant DNA technology for making a medication for treatment in humans. Now, that's not where we stopped. There is something called blood doping. So what is blood doping and how does it improve some athletes' performance? So I want you to go ahead, if you know anything about this, go ahead and answer in the question there. And if you don't know, I don't know is an acceptable answer because you might not be familiar with what blood doping is. So blood doping is using different sorts of medications in order to help improve your performance. Now, specifically recently, which one has been involved is called EPO, which I'll show you what the full name is for that here in a second. But EPO has been at the center of some of the recent blood doping scandals in different sports, because what EPO does is it increases the oxygen carrying capacity of your blood. And so as an athlete, if you think about it, that would be incredibly helpful, right? If you can carry more oxygen, that means that you can have your mitochondria do cellular respiration for longer. And cellular respiration is more effective than fermentation, right? Doing lactic acid fermentation is not going to get you a whole lot of energy. So this is what, one of the things has been used for blood doping. It is dangerous because EPO can actually thicken your blood and lead to a heart attack, but it has been used. And the reason why it's been used over something like steroids, you may have talked about steroids in this, is because EPO is a natural chemical in humans. So we've actually, after we did the development of the insulin-producing bacteria, we also have created one that, a strain of bacteria that does human growth hormone, HGH. Human growth hormone is used for a couple different things. Originally, it was designed so that we could produce it for individuals that have a specific type of dwarfism. So one type of dwarfism is due to a lack of HGH. And so if it's given to them when they're younger, they can actually grow into a normal sized adult. It's also used to combat weight loss in patients like AIDS patients, because HGH is, used for things like muscle mass. This can be used in blood doping as far as athletes. It has been abused because it will increase your muscle mass. And so that is, another one where we started out manufacturing it to treat different diseases, and eventually it was used in some different scandals. Not saying it's widespread, but it has been implicated in some of the athletic scandals as far as blood doping goes. The other one, EPO, so there it is, erythropoietin. It's a mouthful. And this one is, like I said, this one regulates production of red blood cells, so it can increase your oxygen holding capability. It was originally produced for people who have things like chemotherapy, because people who are undergoing chemotherapy have anemia, and also different nutritional deficiencies and things like lung disease, who need this increased amount of red blood cells, because whatever the condition might be, they're not producing enough of those red blood cells. This is actually now not made in bacteria, but it's actually produced in hamster ovaries. I know that sounds funny, but it's the same process. We're producing it in hamsters, but we're able to produce it in sufficient amounts for what we're needing it for. Okay, so we can use biotechnology and genetic engineering to treat or to make medicines to help treat diseases. We can also use biotechnology to help prevent diseases. And this is the other section that we've had some good success in. Because there's 3 different points in our kind of lifespan or reproduction that we can focus on in order to help prevent disease. The first time is before fertilization even occurs. So we can figure out, is a given set of parents likely to have a baby with a genetic disease? Since we know where the genetic markers are for several of these genetic diseases, we can actually test the DNA from both parents and then find out what the likelihood is of those two parents to have a child with those different genetic conditions. So things like cystic fibrosis, Tay-Sachs, there's some genetic hearing loss type diseases, and so on. And so you can see here, since we started doing this sort of genetic screening in 1969, we've actually been able to decrease Tay-Sachs disease by 75% in places like the United States, which is great because if you remember, Tay-Sachs is a fatal genetic disease within a couple of years. So we've been able to do screening. What this allows those parents then to do is make the decision on whether they want to try and have a child together, or to do, use donation or adopt, things like that, in order to limit that possibility of passing on those genes. The next thing we can do is, again, because we know where these genetic markers are and we have the ability to read the genome now, and look for these specific genes, we can find out if a baby is going to be born with a specific genetic disease. Now, this is tested for while the baby is still in the womb, while it's still developing. So this is going to be tested for using things like amniocentesis, where they're going to draw a sample of the amnio fluid, the amniotic fluid from around the baby, because it'll contain cells from the baby. And they will be able to do a genetic analysis and find out, is that child going to have a genetic disease when they're born? And so this is an example of some of the diseases that we can currently detect. So cystic fibrosis, which is one that affects the lungs as far as breathing, and it can limit lifespan. Sickle cell anemia, talked about that one a little bit. That's the one that changes the shape of red blood cells. And if you have both copies of the gene for that, it can be incredibly painful condition. And then Down syndrome, we'll talk about that one a little bit later on. You probably are familiar with Down syndrome. And there are actually quite a few other genetic conditions that we can actually determine on whether the baby has from this sort of sampling. My question is, why would we want to possibly do this? Why would the parents want to know, will the baby be born with a genetic disease? Most of the time, the parents want to know so they can prepare. And I'm guessing that a lot of you, that's probably what you said, something along those lines. They want to be prepared for a child with these conditions so that they're not caught unawares, and they have, what support they need, what equipment they might need, and so on. The last part, as far as preventing diseases with biotechnology, is how likely is an individual to develop genetic diseases later in life. And so this is done through, again, you can have genetic screening done, send in a sample, and they will find out through the genetic markers that we know that are tied to these, how likely it is that you will develop one of these cancers. So breast cancer, prostate cancer, and skin cancer are the ones that we have the best genetic markers for. And so what we can do is, if you know this, what kind of thing can you do that this information might help you with? So if you know that you're likely to develop breast cancer or prostate cancer or skin cancer, what might you do in the future to reduce your risk? Yeah, so you might end up going to the doctor more often. You might end up getting screened more often. If it's something like, if you have a higher risk of skin cancer, maybe you start wearing your sunscreen all the time and wear clothing when you're out in the sun. Do things to prevent, the higher risk of these types of things if you know you have the genetic makeup for it. All right. Last one is curing diseases with biotechnology, specifically what we call gene therapy. And what gene therapy is using the technology to replace faulty copies with functioning copies. So being able to treat or cure that genetic disease by replacing, going in and taking out the bad copies and putting in good functional copies. So this is kind of the holy grail for genetic diseases, if you want to think of it that way. Now, there are lots of different genetic diseases that are being studied for this. This is one of them. So this individual is living in a productive bubble, and he has what's called severe combined immunodeficiency disease, or SCID, SCID. And individuals who have SCID basically have next to no immune system. So they have next to no protection against things like bacterial infections and viruses and so forth. The common cold could kill them, while most of us would just feel, sick for a couple of days. And so some people have called kids who have SCIDs bubble boys or bubble kids, because they do basically live their life in a giant plastic bubble to protect them from the outside world. Now, there has been really good progress made on some versions of SCIDs with gene therapy. So they have done trials with gene therapy with different versions of this severe combined immunodeficiency. And they've actually been able to, as far as we can tell, cure some individuals of their version of this disease. The problem is that there are various different versions of it, depending on how severe it is and what it is genetically that is causing the problem. And it hasn't worked for everyone. So there is progress on this. The other gene therapy that I've seen that has worked has been for a very, very specific type of blindness. Not all blindness, but a very specific genetic type of blindness has worked as far as gene therapy, fixing it by replacing these faulty copies. But it's expensive and difficult to do. So let's look real quick at what the problems are with gene therapy. Why is it that we don't have, gene therapy cures for all of these genetic diseases? Because we've been working on these for quite a while. So the first difficulty is getting the working gene into the specific cells where it's needed. So, for example, if we want to try to work on curing cystic fibrosis, most of the problem with cystic fibrosis is in the lungs. So, trying to get the working gene into the lungs and, staying in the lungs, that is, a challenge. For the blindness one, a little easier because it's very easy to target just the cells in the eyes that need to be targeted. If it's something that's more systemic, like SCID, that one's a little harder, but they were working on it because there are specific ones, like I said, that they could try. But that one is in your immune system. It's throughout your whole body. So that you need to get into the immune cells and the ones that make the immune cells. The next difficulty is getting the working gene into enough cells and at the right rate to create the effect. So it's not enough to get in just one copy. You need to get in, that copy into the cell or the couple of copies, whatever you need for it to work. And you need to get the cell to turn them on and work enough to actually change it. So for SCID, that you've got it turned on so that it's working at the correct level for the immune system. So it's not too low, but it's also not too high. Because remember, if your immune system is in like super attack mode, it can actually be destructive, right? And then you've got a couple of other things. One is the transfer organisms getting into unintended cells. So the thing is that most of these gene therapies are using viruses as basically like their little hypodermic needles. The thing is that viruses don't always go where you want them to. And so some of the SCID patients actually died, it seems, because the gene actually inserted into the wrong kind of cells and inserted so that they turned cancerous. The cells turned cancerous instead of, inserting into the correct kinds of cells and working as they were intended to. So, if we're trying to treat cystic fibrosis, we don't want, those it's intended for the lung cells. We don't want that to get into, say, a liver cell or something like that. And then the last problem is getting the gene regulation to the right level. So that's kind of tied to the, getting it to have a physiological effect, making sure that it turns on when it's supposed to turn on and it turns off when it's supposed to turn off. Because, we're programmed, to have these work correctly, but if you're swapping in the copies, the switches might be not set to the correct spots or levels, and trying to get the cells to actually change that can be a difficulty. All right. The last lecture is going to be on other biotechnology applications that don't fit into agriculture or human health, but are still important as far as technologies that we're using or will be using in the near future.